Dear friends, welcome to this edition of Basic Anatomy in the Allied Health course. Here we cover the Chapter 4, Lecture 1, Nerve Tissue. We will take up a detailed discussion on neurons. In the next lecture, that is Lecture 2, we will cover glial cells. Now, a neuron has characteristics of any other typical cell, but there are certain very special attributes for the cell organelles, primarily because the neuron is designed for excitability, transmission of electrical impulses. To subserve this function, these cell organelles are modified. Now here is a diagrammatic representation of a neuron. You can note the following points. I am not covering very fine detail because the detail of cell structure has already been covered. Only the salient points are being discussed here. Now that is the body or the cell soma, also known as the perikaryon. You can note that there is a nucleus and a nucleolus. Next, in one corner of the cell, there is a small pyramidal elevation which leads to the long process, namely the axon. Now, that large pyramidal hillock is the axon hillock. There are a number of other processes all over the cell. They are the dendrites. But the process that arises from the axon hillock is the axon. You can see the axon going the long axon going into the rest of the areas. Now, those are the dendrites. You can see the branching pattern of the dendrites. Now, the axon has been shown as a long fiber. The moment you say nerve fiber, we generally refer to the item axon. You can see the axon, the cytoplasmic extension, the cytoplasmic membrane that is there around the axon is the axolemma. The cytoplasm that is continuing into the axon is the axoplasm. That means cytoplasm is axoplasm, cytoplasmic membrane is the axolemma. Now, covering this axon is a fat material, myelination, and hence it's called the myelin sheath. These myelin bands are not continuous, they are interrupted in between by the nodes of Ranvier. And now you see at the other end, the axon terminal is there. At the region where the axon terminal, there are no myelin sheath. Now, the cells that produce the myelin sheath is the Schwann cell. Then, in between the myelin sheath, you can see certain gaps where the bare axon is seen. Now, those are the nodes of Ranvier. The area of the axon covered by the Schwann cell, the myelin itself, is called the internode. So, in between the internodes are areas called the nodes of Ranvier. The electrical impulses jump from one node to the adjacent node, and that way this type of conduction is known as a saltatory conduction, which we will discuss in the physiology in more detail. It speeds up the transmission of the nerve impulse. Next, the ribosomes and the rough endoplasmic reticulum are generally well seen, particularly in the region of the axon hillock. Now, these may be seen as dark spots. You can see it in the magnified view also here, rough endoplasmic reticulum and the ribosomes. They are there in this region because they manufacture proteins which are channeled into the long axons. Sometimes they have to travel several millimeters to reach the axon terminals. Next, there are other items generally not seen in light microscopy but worthy of coverage in this discussion, namely the neurofibrils. They are the intermediate filaments which give rise to the shape and the size of the cell. There are microtubules which transport the material from the perikaryon to the axon and into the dendrites, and also vice versa back into the cell body. In addition, particularly in aging neurons, the lipofuscin pigment is also seen. Now, when you look at the different types of neurons, this is a unipolar neuron. The cell body is seen. There is a single process, and that's it. That process is both the axon and the dendrite. But you can see that its variant is a pseudo unipolar cell where it branches into two. That single process. One branch is the incoming sensory electrical signal. The other one is the outgoing electrical impulse. This kind of a pseudo unipolar cell is best seen in the dorsal root ganglion. Bipolar cell. Here the perikaryon is clearly seen. There are only two processes. Outgoing process, that is in terms of the direction of the conduction of impulse, and an incoming process. Incoming process brings in the sensory information, and outgoing transmits the impulse outward. So you can use the word afferent and efferent impulse. Then this is the classic, most often seen cell, the multipolar neuron. The multiple dendrites are shown here by white arrows, and that single axon, which is the longest, is shown by the blue arrow. This is a typical structure of a neuron, and generally we show a multipolar neuron. Now, between neurons there is a wide network, because connectivity is the essence of the function of the nervous system. You can see shown in by the black arrow a junction between the axon terminal of one neuron and the dendrite of an adjacent neuron. That junction is known as synapse. Now, when you enlarge the synapse and see it in more detail, it is a spot where the neurotransmitter is secreted and released, and in a way that is helpful for conduction of the electrical signal. You can see there is a postsynaptic, presynaptic cell and a postsynaptic cell. The terminal end of the presynaptic cell ends in a bulbous ending, and that bulb contains the secretory granules rich in neurotransmitter. A single terminal may have different varieties of neurotransmitter, and it can be secreted as required. Some of the common neurotransmitters that you will see in this discussion is acetylcholine and adrenaline. Now, at the postsynaptic neuron, see there is a synaptic cleft between the pre and the postsynaptic neuron, that white area, and that's a neurotransmitter, that's a synaptic cleft, and on the surface of the postsynaptic neuron you will see number of surface receptors. These secretory granules, the neurotransmitters released, act on these surface receptors and enable the onward conduction of the electrical impulse. Now, this is a complete view of a group of synapses, and this is how a network of neurons is formed to enable a seamless continuity. You can see here marked in red is one neuron, which there is one more neuron in blue and a third one in pink. The pink neuron here, through its dendrites, is in synapse with the axon of the other two dendrites, and you can see its own axon terminal is moving outwards into the rest of the central nervous system. So there are the synapses at the junctions are also, the synaptic junctions are also illustrated, the details as discussed in the previous slides. Now, the pyramidal cell and Purkinje cell are two classic examples of neurons. We have seen the Purkinje cell in one of the previous diagrams, especially when the Nissel's granules were discussed. Now we are showing another cell, namely the pyramidal cell. The pyramidal cell is one of the largest neurons we know of, and you can see the flashing arrow shows this pyramidal cell. These pyramidal cells are usually triangular in appearance in H&E preparations and other immunofluorescent preparations. They are really large cells appearing triangular, but they are actually pyramidal in three-dimensional view. The dendrites are there in all the three edges of the three corners of the apices of the triangle, but the single large and a very long axon arises from the basal aspect of the cell, that is that area opposite to the apex, and that goes down into the central nervous system, and often it travels very long distance. The pyramidal cell is a classic example of a motor nerve cell. Thank you. That was a discussion on the overall features of a neuron.